Good morning, everyone. Welcome to the 2024 NARO-JIRCAS-FFTC International Symposium Day 2. To begin with third sessions, we will want to start from now. Mr. Ken Nakamura from NARO will be the moderator of the sessions. Mr. Nakamura, please everybody give a round of applause.
Good morning, everyone. Let's start this third session of other technologies for reducing GHG emission and rice heavy metal adsorptions. And Dr. Noriko Yamaguchi from NARO is the co-moderator. And the first speaker is Mr. Mohd, Zafrul, Arif, Radhi from MARDI. Let me introduce his simple careers. He belongs to the Malaysian Agricultural Research Development Institute, and he got a PhD. He got a degree BSc in University Cabasang, Malaysia, and at least he's working on the development of bioremediation product to solve the heavy metal pollutions. And his title of this session, title of this speech is Harnessing Microbial Power and Biofertilizer Solution for Heavy Metal Pollution in Rice Field. Mr. Radhi, please speak.
Thank you, Dr. Chairman, for introducing me. Konnichiwa. Good morning. I am Zafrul from MARDI, Malaysia. Okay. I'm very glad today to present to all of you for this third session, which is Harnessing Microbial Power Biofertilizer Solutions for Heavy Metal Pollution in Rice Fields. Okay, first of all, I'm very sorry because actually I should be presenting physically, but for some reasons, technical reasons, I couldn't make it there. But I am very fortunate because FFTC give me the chance to present online. Okay, these are my presentation outline for today. First, we will look into the introduction a bit, and then about contaminated paddy soil. And then we will move on to bioorganic smart fertilizer. This is the biofertilizer that we are developing, okay, to address the issues of this arsenic and cadmium contamination in paddy soils. Okay. And then some research findings, and then additional info on bioremediation, and then we come to conclusions and reference. Okay. So actually one issue that requires careful consideration and prompt action is heavy metal pollution for rice crops. Okay. Because in Malaysia, rice is, as in other Asian countries, are the staple food, and then pollution in this food chain is very serious and dangerous. So we have to address these issues, and in MARDI we have some research going on into tackling these heavy metals issues and also the greenhouse gases issues. Okay, for me, I am from the program of fertilizer technology, so I'm a research scientist there. So basically, we develop fertilizers. For me, it's biofertilizers. Okay, and then there are other centers in MARDI, which is biodiversity and environment. That one tackles mostly for the GHG, greenhouse gases and mitigation and so on. Okay. So in this method, we are trying to use this bioremediation. Okay? As you all know, bioremediation is a procedure that uses microorganisms, plants or enzymes from plants or microbes to detoxify contaminants in the soil and environment. So this is one way to cope with heavy metal contamination, specifically in this paddy soil. Okay. What is a contaminated paddy soil? Okay, contaminated paddy soils refers to the soil in rice fields which has been polluted or altered by presence of harmful substances such as heavy metals, pesticides, chemical fertilizers, or industrial waste. Okay, it gives negative effects on soil quality, crop productivity, and most important, the human health. Okay, and it can also affect the quality and safety of food crops, making it an important environmental and public health issue. Okay, here comes the biofertilizer that we are developing, which is the bioorganic smart fertilizer. In this bioorganic smart fertilizer, it contains organic and biological materials because it is from, we have the source of the biofertilizer is from the NPK, nitrogen, phosphorus, potassium source is from the organic source. And then it can promote soil health, increase crop yield, and reduce the negative impact of synthetic fertilizers on the environment. Okay. Typically, it contains a range of beneficial microorganisms. In this case of biofertilizer that we are developing, we have two strains of bacteria, beneficial bacteria. Okay. And then it can support the growth and development of plants and enhance soil fertility. Okay. And this fertilizer is different from traditional fertilizers. Okay, because we know that traditional fertilizers made from compound synthetic fertilizers, high level of NPK, but this one it only provides necessary nutrients for plant growth, and it doesn't harm plants, animals, and human health. Okay, this is the works that we have done. First, we did the bioprospection of the suitable bacteria for arsenic and cadmium bioremediation. Okay. Because we have to search for this bacteria to be incorporated into the biofertilizer. Okay. So we did the bioprospection in Malaysian soil, in Semenanjung, Malaysia. Okay, way back in October 2021, we chose the granary areas in Pasir Putih, Kelantan, Seberang Perai, Penang, and also Kedah. From the 21 soil samples we collected from 5 distinct places in Peninsular Malaysia, we managed to isolate and screen for arsenic and cadmium bioremediation microorganisms. And then we successfully identified 40 bacterial isolates with resistance to 50 milligram per liter of arsenic and cadmium. This is a 50 ppm. So all the bacteria can withstand the arsenic and cadmium up to 50 ppm. 10 isolates were chosen for the most promising microbial screening technique for arsenic and cadmium bioremediation, 5 of which were arsenic resistant bacteria and 5 of which was cadmium resistant bacteria. So we shortlisted it according to the performance of the respective bacteria. Okay. And then on the right hand side is the picture of when we do soil sampling in Tanjung Karang, that one in Tanjung Karang. Okay. So it involves sample preparation and then the metallite solutions, and then we did isolations of the resistant bacteria. We isolate it in media, different media, and then we see the resistant pattern. We use this method, this diffusion method. So we see the MIC, minimum inhibitory concentration. Okay. From that, we can screen the potential bacteria, and then we send it for identification. We use the PCR, and then we use the DNA fingerprinting for bacteria. Okay, as the results, we found we use the 16S RNA technique, and then in figure 1 you can see AS1, CD1 and CD3 with the most highest potential and base pairs around 1500 base pairs. This is the most potential isolates that we found from the screening, and then we send for sequencing. However, CD3 we do not select it for further work because it is found to be a pathogenic strain to human. Okay, this is important for developing any kind of biofertilizers, because in my country also we are facing this problem whereby they develop biofertilizers, but they use the use of microorganisms. Sometimes they use the microbes which is harmful to either human or animals or other plants, because microbes, not every microbes is a single purpose. I mean, it can be good and it can be bad in something. So we have to choose, really choose only the beneficial microorganism. Okay? So that's very important, because there are microbes that can do wonders in soil, that can give high yield to crops and so on, but at the same time is harmful to human. Okay. Okay, we did a phylogenetic analysis and isolates arsenic 1 and CD1. We found out that later it is closely related to Bacillus megatherium, strain ATCC 14581, and Bacillus proteolyticus, strain MCC 1A00365. Okay, so the two chosen probable metallite removing bacterial isolates were found to be closely related to Bacillus megatherium and Bacillus proteolyticus. Okay. So this is the results of phylogenetic tree. Arsenic 1 is here, and cadmium 1 is here with the grouping of Bacillus proteolyticus. Okay, this after we have found the bacteria, suitable bacteria, and we formulate it into biofertilizers. Okay, So this is the prototype of biofertilizer that we developed. It is in tablet form. Okay. So we name it as cat. Okay. So basically the NPK is not that high because it is organic based. And it also have other organic matter is more than 50%. Because in Malaysia, in our standard, if we want to declare the fertilizer as an organic biofertilizer or organic fertilizer, the organic matter should be more than 50%. So these are the trials that we are conducting in Sungai Balang Muar, Johor. Okay, this trial is still in going. Okay, we have 8 treatments. Okay, this is the RCCAT is the product name. Okay, so we aim here is to do testing because as we know, bioremediation carried out by the microbes, okay, it can oxidize the arsenic, for example, from arsenic 3 to 5, but into less toxic form, But after sometimes there are other microbes also in the soil, right? So as we know, bioremediation by microbe, the heavy metals we cannot diminish it, or we cannot totally dissolve it by microbe action. So in our study, we also include this Kajanus kajan. Kajanus kajan is type of phytoremediation technique, because Kajanus kajan is, if you know, is a pigeon pea. In India, or is very popular. So this can be a phytoremediation for these heavy metals. Okay, so the idea is that we use the microbes, the biofertilizer with microbes to convert it to less toxic form for the arsenic and cadmium. At the same time, we use this Kajanus kajan to absorb away it from the soil. So we can remove it from the soil and let it be in the safe place. Maybe we can incinerate it, and then they always do it like they include in bricks, because in bricks to do construction. Okay, so the ashes they mix in bricks and do construction, so that the heavy metals will be fixed there, so cannot pose danger to others lah. Okay. So this is the sample preparation in our lab. You do digestion for the ICP analysis. This is the production of RCCAT biofertilizer. We use the tablet press machine. Okay, this is just for small scale. If for large scale, there are bigger tablet machine. Okay, this machine can produce about 15,000 tablets per hour. Okay, this is the kajan that I said, the plant. We tried several times to make it, to domesticate it. I mean, because this plant is not very commonly planted in Malaysia, okay? But it can grow well because of the climates. It likes the hot climates. Okay? So we tried also in the plant growth chamber. Okay, this is just the reference because reference value for arsenic and cadmium. In our country also we have a different. Sometimes they refer to Dutch list, sometimes from the university, but generally for our Department of Agriculture is 1 ppm for arsenic and cadmium in the soil. Okay, these are the 3 rice growing areas with high content of arsenic and cadmium in Peninsular Malaysia as published. But I believe there are more because the one that I did in Sungai Balang also we found twice the amount of arsenic there. So the main area there is Pasir Puteh in Kelantan and Krian in Perak and also Kubang Pasu in Kedah. It's all in Peninsular Malaysia. Okay, these are just additional info on what is bioremediation. Okay, this is by bacterial cells. There are some processes which can do like biotransformation, biosorption, bioaccumulation, bioassimilation, bioleaching, bioprecipitation. Okay. But for arsenic bioremediation mechanism, okay, it involves oxidation and reduction. Some microbes can reduce the less toxic arsenic into arsenite through the action of enzyme arsenite reductase. While arsenite is more toxic, it is often excreted by the microbes, reducing its presence in soil. Others are arsenite oxidation also. There are microbes which can oxidize arsenite into arsenate, which can then precipitate as insoluble mineral forms like iron arsenates, and it decreases its mobility and toxicity. As for the cadmium, bioremediation mechanisms is mainly biosorption, okay, and then bioaccumulation. Bioaccumulation is that some microbes can actively take up this cadmium ion and sequester them inside their cells. Okay, and then precipitation and mineralization also. There are these microbes can do. Okay, like they react with cadmium to form cadmium sulfide, and cadmium sulfide is a insoluble compound which can precipitate out of the soil solution and become immobilized. This is important because when it's immobilized, it is not toxic, less toxic. Okay. So this is just the bacterial cells. Okay. Okay, this is the conditions in the soil where when the microbes act. You can see. Okay, this is also the process. This is the process of phytoremediation. Phytoremediation They have this volatilization, transpiration, phytoaccumulation. This Cajanus cajan mainly is good for this phytoaccumulation because it is a hyperaccumulator of this cadmium, where the cadmium will be stored in plant parts such as the roots and shoots. Okay, and then when we plant it, we can remove all the plants from the soil so that we can take out all the also the toxic compounds there. Okay. So as a conclusion, we found two these bacterial species shown to be promising bacterial strains for arsenic bioremediation and cadmium bioremediation. So we are still continuing our works to test it on the field, okay, and then hopefully we can get good results because it is working on the lab scale, and then on the field it is showing some good signs. So we are still waiting for the results. Okay. These are my reference. Okay. So I would like to acknowledge my research team as stated here, and also MARDI Management and Research Grant Provider for Rancangan Malaysia ke-12, RMK 12, PRP 515, and all parties indirectly involved. Also, thank you for FFTC NARO-JIRCAS for allowing me to present here today. So, thank you. Thank you very much.
Thank you, Mr. Radhi. Is there any questions and comments, please?
Good morning, everyone. I am Dr. Sanjay Swami, representing Central Agriculture University from India. And many thanks to Dr. Radhi from Malaysia for a very insightful presentation on bioremediation. Dr. Radhi, my query is that you are using this different species of bacteria along with Cajanus cajan. So how you are implementing or executing your treatment? Because Cajanus cajan is a huge tree. So how you are growing this in paddy soils? It is not possible.
Okay, actually our aim is that first we apply in Malaysia, if I'm not sure in other countries, in Malaysia, if the regulation, if the soil or we found out the heavy metals upper the limit, up to the limit, okay, over the limit, so actually they have to stop the activity of paddy planting at once, and then they have to recover back the soil, okay. So that is the point when we will use this what we call the biofertilizer. We will apply it, and then because it also, besides it have beneficial microorganisms which can bioremediate, it also have this extra nutrients that can enhance the soil fertility, okay. And then after we apply the fertilizers, we will plant out this Cajanus cajan. Cajanus cajan is fast growing, okay. in
It's
about
fast growing, but for how long time
Yeah.
you are taking for bioremediation? That is
Yeah,
also one of the big question, that because if you are taking 5-6 years at least, then you
no.
have to devote that paddy
No,
soils for
no,
remediation purpose.
5 to 6 years. Actually, it is like 2 months, 1 to 2 months. It already, because we just want it to have the shoots and roots growing, and then we will remove the plant. We will not wait until it bear fruits and something.
No, but in 1 or 2 months it is not possible. That is my observation. That one advice I can give you, that instead of this caseous casein, you can use alongside of your this bacterial strain this vermiremediation techniques. That is also gaining very momentum. You can use
Yeah.
the local strains of your worms, vermi, and then can just screen out the potential species. That you can try. Thank you.
Yeah. Yeah. Okay. Thank you for suggestion.
Thank you. Time is up. And thank you very much, Mr. Radhi. And please give him a big applause again. Thank you.
Thank you.
Next speaker is Dr. Yu-Wen from USDA ARS Dale Bumper National Rice Research Center. And he got a PhD from Paddy University, and he is the plant pathologist with over 30 years of successful experience in plant genetics, pathology, molecular biology, biochemistry, and microbiology. And the title of the presentation is Impact of Multiple Dry Downs on Rice Yield, Processing Grain Quality, Trait, and Greenhouse Gases. Please, Dr. Jia. And next speaker is Dr. Yu-Wen Lin. And she is from Taiwan Agricultural Research Institute, and she got a PhD from National Changshu University. And she's the soil chemistry, she's majoring in soil chemistry, soil plant analysis, and soil fertility and soil contaminations. And please, Doctor.
Okay, thanks for Dr. Nakamura's introduction. And good morning, ladies and gentlemen. I'm very pleased and honored to have the opportunity to participate in this symposium and make a presentation. Now I work at Agricultural Chemistry Division of TARI MOA, and I am the head of soil analysis laboratory, also head of an ISO 17025 accredited laboratory. And I'm also the organizer of the development of low carbon farming technologies project of MOA. Next, please. And this presentation includes two parts. The first is about research and integrated control procedures to lower the risks of rice with a cadmium or inorganic arsenic concentration exceeding the regulatory limit. And the second is about the current situation of developing low carbon farming technologies for rice production in Taiwan. Next, please. And let me start from the research on relationships between cadmium concentrations in rice and loss in arable soils. And to ensure food safety in Taiwan, the Ministry of Health and Welfare has established regulatory limits for food metals. In addition, the Ministry of Environment sets regulatory limits for heavy metals in soil on food crop farmland. However, due to diverse geological conditions in Taiwan, and also due to some anthropogenic activities, in some areas, the concentration of heavy metals in the soil is higher than the background values of Taiwan. So in some cases, cadmium concentrations in rice, peanuts, or garlic exceed regulatory limits, even though the cadmium concentration in the farmland is below the regulatory limits. From 2005 to 2009, and from 2016 to 2017, field trials were conducted at 23 fields in 8 sites in western Taiwan to clarify rice cadmium absorption characteristics. The experiments were performed on farmland that had been polluted by heavy metals in the past or had a high natural content of cadmium in the soil. Okay. So some of these fields were polluted by irrigation water contaminated by illegally discharged wastewater from electroplating, smelting, or pigment industry. Therefore, the soil cadmium concentrations at each field were not uniform. And calculate bioconcentration factors of cadmium in different rice cultivars indicated that cadmium accumulation in the Japonica species is lower than in the Indica species, with cultivar Taidong number 30 having the lowest cadmium accumulation capacity. And the soil plant transfer model of cadmium in different rice cultivars have been established using testing data of soil and brown rice collected from field experiments. And these models allow soil cadmium concentration, pH, and CEC to predict cadmium content in brown rice. And according to the regression coefficient of the soil plant transfer models, we found that both pH, soil pH, and CEC, soil pH and CEC have negative impacts on cadmium concentration in rice. And in 2017, a field experiment was conducted in central Taiwan to investigate the effect of water management on cadmium availability in soils and its accumulation in rice. 2 water management treatments were tested, that is conventional and prolonged flooding. The main difference between these 2 treatments is water management after 50% heading of rice. In conventional management, after 50% heading of rice, and AWD management was adapted, while in prolonged flooding treatment, after mid-term drainage, a flooding condition was maintained until seven days before harvest. And the results indicated that prolonged flooding could effectively reduce the cadmium concentration in brown rice compared with conventional practice. And since 2016, management methods to reduce cadmium concentration in rice have been promoted on high-risk farmland that once produced crops with cadmium concentration exceeding the regular limit. And the main measures taken are planting low cadmium accumulation cultivars, liming of the soil, and prolonged flooding. And based on the research and the promoting results, integrated control procedures of cadmium in rice was established. In 2020, technical guidelines for the safety management of crops on high-risk farmland that once produced crops with cadmium concentrations exceeding the regulatory limit were proposed. The suggested measures depend on the soil testing results. First, if the soil pH is lower than 6.5, we evaluate the lime requirement and lime the soil to raise the pH to higher than 6.5 and lower than 7.5. After adjusting the soil pH to above 6.5, we suggest rice cultivars according to the predicted cadmium concentration in rice using the soil plant transfer models developed. When the predicted rice cadmium concentration is high, we suggest farmers plant low cadmium absorption cultivars and also adopt prolonged flooding water management. And in addition to recommended cultivation techniques, in Taiwan, the government also has corresponding policy incentives and restrictions to encourage farmers to adopt such cultivation methods. The restriction is once the concentration of heavy metals in crops exceeds the regulatory limit, farmers must follow the farming methods recommended by agricultural research institutions before planting edible crops. Otherwise, food crops cannot be grown in such fields. And the incentives are, first one, the government subsidy for the cost of lime application. And second one is if farmers follow the farming methods recommended by agricultural research institutions, but still produce crops with heavy metal concentrations exceeding regulatory limits, the cost and the cost of crop removal and burning and the year losses will be subsidized by the government. And through the implementation of these management measures, instances of rice cadmium concentrations exceeding regulatory limit have been rare in recent years. Okay, the next topic is research and the measures to reduce risks of rice with inorganic arsenic concentration exceed the regulatory limit. And from 2020 to 2021, water management experiments with 2 cultivars were conducted in 4 sites in Taiwan. The cultivar used were Taichung Xian number 10. It's an indica species, and Tainan number 11. It's a japonica species, and the water management tested was conventional, prolonged flooding, and AWD. And the results showed that the inorganic arsenic accumulation capacity of cultivar, Taichung Xian number 10, was significantly lower than that of Tainan number 11. And the inorganic arsenic concentration of the second season brown rice is about 2/3 of that of the first season rice. And what's more, AWD during the late grow stage reduced the inorganic arsenic concentration of cultivar Taichung Xian number 10 by an average of 15.6% compared with conventional treatment, while the prolonged flooding increased the inorganic arsenic concentration by an average of 17%. And as we know, although published articles indicate that many factors may influence arsenic accumulation in rice, but only the effects of rice cultivar and only the effect of rice cultivar, soil moisture, and the iron oxide content are apparent. Of these 3 factors, only rice cultivars and soil moisture can be controlled by farmers. Therefore, planting an indica species and adopting AWD irrigation are currently the main management measures to reduce inorganic arsenic concentration in rice in Taiwan. Okay, the last topic of my presentation is the development of low carbon farming technologies for paddy rice. This slide shows Taiwan's greenhouse gas emissions from 1990 to 2022. In 2022, agriculture sector accounted only 1.11% of greenhouse gas emissions among net emitting sectors. However, it's very low. However, no one is an outsider in the face of climate change, and the agriculture sector must also strive to achieve net zero emissions. And the primary sources of greenhouse gas emissions from the crop production sector are methane and nitrous oxide. Methane emission from rice production and nitrous oxide emission due to the nitrogen application. So for crop cultivation, the GHG mitigation research conducted in Taiwan focuses on reducing the methane emissions from paddy fields and the nitrous oxide emissions due to nitrogen application. For reducing methane and the nitrous oxide, research focuses on solving the main factors causing methane emissions, the presence of organic matter, and the anaerobic environment in the paddy fields. And for the nitrous oxide emissions, nitrogen application is the main causing. So the three major research topics were rice straw removal and the proper handling, and AWD, and also enhance nitrogen use efficiency. And in 2023 and 2024, the effectiveness of rice straw removal in reducing methane emissions from paddy fields has been evaluated in many experimental fields. The results showed that rice straw removal can reduce methane emissions from paddy fields by 10% to 66%, and does not significantly affect rice yield. And the removed rice straw can be used to investigate value-added applications such as producing fuel rods, biochar, straw mushroom culture media, and biodegradable tray liners for example, for pineapple or egg cases. And the AWD water management recommended by IRI was also tested in Taiwan in 2023 and 2024. The results in 2023 showed that using AWD can reduce methane emissions from paddy fields by 21.5% to 66%, but it also lowers rice yield in most tests. To reduce methane emissions without reducing rice yield, different AWD drainage strategies were tested in 2024 to propose optional guidelines tailored to local conditions. And although it is well known to soil scientists that nitrate formula fertilizers should not be applied to paddy fields, however, due to a lack of awareness and consideration of the convenience of application, some farmers in Taiwan still prefer to use nitrate formula fertilizers in paddy fields. And in 2024, a field experiment was conducted at TARI to evaluate the effects of nitrogen foam on nitrous oxide emission from paddy fields. Two experiments were conducted at TARI to evaluate the nitrous oxide mitigation effect. The first one is I just mentioned, to test the nitrogen formula. And the second one is to evaluate the slow released nitrogen. And the results showed that using ammonium instead of ammonium nitrate mixture can reduce nitrous oxide emissions by 83.2% and increase grain yield by 18.7%. And using slow-released nitrogen instead of conventional nitrogen fertilizer can reduce nitrous oxide emission by 54.7%, and increase grain yield by 3.7%. I think that restricting the application of nitrate fertilizers in paddy fields through the fertilizer purchase registration system of MOA should be one of the most feasible ways to reduce nitrous oxide emission in paddy fields. And conclusions. First one, integrated control procedures for cadmium in rice has been established in Taiwan, including planting the Japonica species, liming, and or increasing the flooded time. Second, through the implementation of these management measures, instances of rice cadmium concentrations exceeding regulatory limits have been rare in recent years. And the third one, planting indica species and adopting AWD irrigation are currently the main management measures to reduce inorganic arsenic concentration in rice in Taiwan. And the fourth, restricting the application of nitrate in paddy fields through the fertilizer purchase registration system should be one of the most visible ways to reduce nitrous oxide emissions in paddy fields. And the last one, management SOP tailored to local conditions must be formulated to reduce methane emissions without reducing rice yields before fully implementing AWD irrigation method in Taiwan. So that's all my presentation. Thank you.
Thank you, Dr. Lin. Is there any comments and questions? Please.
Thank you, Dr. Yu-Wen, for your very informative presentation. I have a couple questions. First, on the arsenic, you show the first crop and second crop, right?
Yeah.
Yeah. Could you show that slide?
Okay.
I think yesterday we discussed that for arsenic absorption by rice, one is effective, the influencing factor, one is the redox potential. Oxidation would have fixation of the arsenic, reduce it. So AWD would be effective.
Yes.
And then also the temperature that would increase the evapotranspiration. So you see here from our first crop.
Yes.
First crop, the arsenic is higher than the second crop because our first crop is from a cooling environment. Until harvest, it's very hot. So it's so the temperature is higher, and yeah. And then the second crop, it's from warm. Usually it's from July to August planting, and then harvesting in after October or so. So it's cooler. So I think that shows the both. I mean, also approve that. So
yes.
So that's one point. It's just a comment. But I noticed that the AWD you applied here, you show it start from right after transplanting. The water regime for the arsenic treatment.
This one?
Yeah. So you see, when did you start the AWD?
Because
for how long?
This AWD not fully follow the ERI proposed. We do not, we re-flooding when the water level low down to the ground level.
Oh, it's not minus 50, it's
not-
to 0.
Yes.
Okay, but then when did you start it? It's how many days after this seed transplanting, right? It's a transplanting. It's not
just
direct
from the tilling stage. No. AWD yes， 就那个开始分裂以后 ，tilling stage。
Really? Okay. So start teetering.
对 ，yes。
Okay. So how many days after transplanting?
It's different.
Okay. No matter. I think, and also the other experiments for your the last one, you show the AWD experiments. You started the drainage 2 weeks after transplanting. I think compared to others, maybe it's too short. So maybe you should try.
Yes.
Try different, maybe longer.
You mean the AWD adopted in the GHG mitigation project?
Yeah,
Yes.
I saw your picture shows 2 weeks after transplanting.
This just for the heavy metals, and such experiments conducted many years ago. And in the GHG mitigation project.
But otherwise, you show that picture. You have the yields, and then the water regime. I think that one, in the greenhouse gas emission.
This one.
Yeah, I think the water. You show the water. You show the diagram, how you do the drainage. Previous one.
This fully follow the
So you said first 2 weeks after transplanting. So maybe try a little bit longer.
yes.
Yeah, 3 weeks.
So
yeah.
this year we try to test the different control measures in different areas to test the GHG mitigation effect, and also not to reduce the yield.
Okay. Sorry for taking so long. Yeah, okay. The last one, it's really the question. If you have both contamination of arsenic and cadmium, what are you going to do? That's a question for you. Thank you.
I think if the best suggestion maybe farmers should to change the crops they planted, maybe not to cultivate the rice, maybe because we do the heavy metal research, and not only on the rice, also many vegetables, crops, and the peanuts were included. So if the farmer cannot plant rice, we will suggest them to choose another crops to cultivate. Yes.
Thank you, Dr. Lin. Time is up, and please give her a big applause again. And thank you very much. Then let's close the sessions.
Thank you very much, Mr. Nakamura, and all the presenters. So now we are going to have a little break time. Coffee and tea will be prepared outside at the conference room. Please come back to your seat by 10:50. Thank you.